friends, welcome back to the journey of microbiology. We already discussed the two chapters, covered the two chapters in microbiology, and now today we will see the another chapter in microbiology called the immunity. Okay, we will study the immunology basically. Now, you came across so many infectious disease, okay, you may have contact with the infectious persons, but your body is not suddenly responding, okay, your body is not suddenly responding towards the external infectious agent. Why? I am telling you why. Because our body have the capability to restrict the pathogenic, we can say, manifestations, pathogenic manifestations. Our body have the capability to fight with the external, or we can say the foreign particles. Okay, so today we will study about the one aspect of immunology, that's called the immunity. That's called the immunity. Immunity. Okay, now, so before discussing the immunity, we first see our lecture outline, what things we will cover in this lecture. So as you are seeing, first we will deal with the what is the meant by immunology. Okay, what is the meant by immunology. Okay, then we will see the immunity. Okay, what is immunity and their types. Okay, for ease of study, for sophistication of study, the immune system is classified, so we will see also the classifications. Then we will see how the immune cells, okay, you know that each and everything in our body is ultimately contributed, or we can say the channelized by the basic fundamental unit, that's the cell. So how immune cells come into the picture, I mean come into our body, and how they are formed, how they are processed, what are their types, okay, where is their originations, from where they are originated, everything we will see in the under the topic of development and processing of lymphocyte. Okay, lymphocyte. Then we will see about the antigens. Okay, the antigens. Then development of cell mediated immunity. Clear? Then we will see the development of humoral immunity. Okay, then about the natural killer cells, then cytokinins, okay, and you know the immunizations, okay, we can say in another word vaccination. Then we will see the abnormal response, okay, abnormal response of our immune system. The first we will see under this abnormality is allergy and immunological hypersensitivity reactions. Then some of the immune deficiency diseases and autoimmune diseases. Okay, these are our lecture outline. So first we will go with the understanding the meaning of immunology. Okay, understanding the meaning of immunology. Immunology. See, if you want their literal meaning, then you understood, you can break down this word into two parts called immune and logy. Immunes means something which resists external agent to come in the body and show their phenomenon, so their, we can say, the process, and logy is study. So basically immunology is a branch of medicine, or for more specifically if I am talking, then it's a branch of infectology. It's a branch of infectology. Under this branch we will deal with understanding the basic function, okay, I am writing function like this, F, and function of immune system, immune system, okay. Then we will see about their development, how immune cells are developed, okay, development, and their abnormalities, or we can say the when the immune system is failed to functioning, then how our body is reacted. That's also come under the study of immunology. So we can say the abnormal, okay, abnormal status of immune system, okay, the abnormal status of immune system, function of immune system, and development. Clear? That's a three aspect we will deal under the branch of immunology, okay. It's a broad branch, okay. It not deal with only these three things, it's also deal with some of the advanced things, like you know that immunology, okay, it's a cell, immune cell, then cell must have the nucleus must have the genetic materials, and there are diseases where the immune nucleus genes is altered, okay, it's altered or mutate. So that's called the immunogenomics, okay, that's called the immunogenomics. Write down this word, you will come across in the textbook, immunogenomics, okay. This immunogenomics is basically study of the immune cell genetics, Probably you heard about the cancers, okay. In the case of cancers, what happened, the immune cells which fight, basically the lymphocyte, T lymphocyte, B lymphocyte, we will see about these things. So how this T lymphocyte, B lymphocyte genetic material is altered in the case of cancer, that's everything is studied under this particular branch of immunology, okay. Keep in back of your mind that immunogenomics is the sub branch of immunology, okay. Immunogenomics is a sub branch of immunology, and under the immunogenomics we separately only focus to understand about the immune genetics, okay. Hope you understand this. Now after understanding the immunology, okay, we will see the branches of immunology. Now why branches are classified, why we have to study so many things. See, branches or classification is very much necessary, okay, it's a very much necessary, as we are seeing that our body is very simple, okay, but if you pay your keen attention, you will gonna surprised that our body has so many functions, they are doing so many functions to stay us alive, okay. So for understanding all those functions is, we have to classify the things, okay, we have to classify the things, separate the things, and then study, okay, because there are drugs, there are medications, okay, which work particularly on one particular function of our body, So for immunology, can you tell the name, which kind of drug we are using? We are saying those category of drug is immunotherapy. Under the immunotherapy, we are using so many things, and vaccination is one of them, okay. So now come to the point, we are dealing with the branches of immunology, okay, branches of immunology, okay, branches of immunology. Now under these branches of immunology, we already discussed one branch, okay, and that branch is called immunogenomics. Immunogenomics, okay. Now immunogenomics is basically the study of immune genetics, okay. The another branch in the immunology is called Immunology. The term is bit long. Psycho, neuro, immunology. Now, my friends, you understood this by seeing this term. You understood it's basically the study of immune functions in our nervous system. Okay, in our nervous systems. Okay. Now, one thing you keep in your mind that not always the immune cells are functioning towards the some of the known diseases, like some of the known familiar diseases, cancers, the bacterial infections, all these infections. No, there are disorders, okay, where these any particles, any foreign particles is not involved. Okay, our own cells are working against us. On that very disease, immune system are also functioning. So in the case of some of the disorders, okay, like the some of the Parkinson disorder, we can say some other neuropsychiatric disorders in which the pattern of immunological cells is altered. The pattern of immunological cells is altered. Clear? Now, the another branch of immunology is called diagnostic immunology. Now, see, this is very interesting branch. Diagnostic immunology. See, you know that your body possesses two things, the antibody and antigen. Okay, now antibody is represented like this. Okay, it's a antibody, okay, and the antigens we can represent simply by the solid diagram. Okay, so one of the bases of immune function is interaction between the antibody, okay, and antigen. Interaction between the antibody and antigen result into the either our body win or the infectious antigens win the match. That's the whole story. Okay, so what happened? These antibody antigen reactions becomes the basis to diagnose so many diseases. Okay, may you heard about the ELISA test. May you heard about the ELISA test. ELISA test, see, ELISA test is basically the enzyme linked immunosorbent assay. Enzyme linked immunosorbent assay. Under this ELISA test, what happened? Some of our body fluids, basically the blood is taken, okay, and then we performed the antibody antigen reaction, antibody and antigen reactions, and we will see which kind of antigen is present. Okay, this test, ELISA test, is a diagnostic test for exactly the HIV, or we can say the AIDS. In the case of AIDS, this ELISA test is employed, or this ELISA test is employed. Now, the another test, you know, that's called the PCR. That's called the PCR, polymerase chain reaction. Polymerase chain reaction, and you know the RT-PCR, one of the form of PCR which employed in the case of corona. In the case of RT-PCR, what happened? We will take one gene, okay, and we polymerize that gene, okay, and see if the minute amount of antigen is present of the infectious disease. So by the PCR test, we can multiply them in a millions of the copy and see clearly, okay, this disease is found. Clear? That's called the PCR. So all the ELISA test, PCR test, and there are so many other staining techniques. Okay, we have some of the staining techniques. All comes under the sub branch of immunology, that's called the diagnostic immunology. Clear? Now, we have another you written these things. Now we have another branch that's called the reproductive immunology. Now I am going to just shortly describe these branches. That's called the reproductive immunology. No, this is the study of the reproductive immune system. Clear? Now we have the another branch called the nutritional immunology. Nutritional immunology. Clear? Under this nutritional immunology, we will establish and already established. There are so many researchers who established that nutrition play a pivotal role to either enhance your immunity or we can say compromise your immunity. Either you are immunocompetent or immunocompromised by the nutritional deficiency or we can say the adequate nutrition. Clear? That's one sub branch of immunology. Then we have immuno-oncology. Then we have the immuno-oncology, or we can say the immuno-oncology. See, this oncology is basically the study of, it's the study of the cancer. It's the study of the cancer. So this branch specifically deals with the study of cancer in respect with their immune changes, their immune cells function changes. Okay, that's called the immune oncology. So these are some sort of branches of immunology which I discussed. Okay, so if I summarize these branches, we will see in the nutritional immunology, immuno-oncology. Okay, then we see the diagnostic immunology, reproductive immunology. Okay, these are some of the branches of the immunology. Now after understanding the immunology, branches of immunology, we will see the immunity. Now, so understanding the immunity and immune system, these are the two things. Okay, not the separate thing, they are the same thing, but immune system shows compromised organized body structures, okay, which help in the immune functioning, or we can say which deals with the immunity, Immunity literally means resisting, okay, resisting the body to fall down towards the disease, that's called the immunity. So we see the immune system, You understood that immune system is the one which deals with the foreign particles, we can say the cellular debris, okay, any infectious agents, microbial diseases, viral diseases, every diseases these systems are involved in dealing with. Okay, now immune systems is, we can say, it's either, if we talk about their type, if we talk about their classification, it may be either inborn, okay, or it may be either acquired. Okay, it may be inborn or it may be acquired immunity. See, what is meant by the inborn? Inborn means, see, we have hereditary line, okay, we follow the genetics pattern, okay. So these genetic patterns, we are not inherited only some of the characters from the parents, like the color, okay, the height and all that stuff. We are also acquiring, we are also inheriting some of the immune cells, okay, we can say the immune systems. Okay, now these inborn system is also called innate. Okay, innate means since birth, we can say it's the innate, immunity. And acquired immunity is those kind of immunity which we acquired in our, like we can say, in our life pattern. Okay, in our lifetime, whatever things we acquired, we adopt. So this is called the acquired immunity. So in another word we can say it is also called as the adaptive immunity. Adaptive immunity. Okay, so now this innate immunity we understood it's come from since the birth, okay, and acquired immunity are the adaptive immunity which we acquired during our lifetime, okay. Now this inborn immunity, they are called non-specific type of immunity. They are called as non-specific immunity. Why? There must be underlying reason, that's why they are called non-specific. They are non-specific, see, and they are called specific. They are called specific. See, non-specific means they are like a blind, okay, they are a blind. Whatever comes, they first go. You know, like anything, any foreign particles in the body comes, they blindly go to there. Why? Because they are not driven by any memory. They are not driven by any memory cells, okay, and they are the one who is work as a, we can say, like barriers. They are working, these inborn immunities, they are working as a barrier kind of immunity, okay, and because they are going blindly, they are not specific, okay, they are not specific towards the specific pathogens, specific kind of bacteria, viruses, microbes. That's why they are called non-specific. Whereas in the case of acquired immunity, they have the memory cells. First of all, they are driven by memory cell. Means in the first time, this kind of immunity, they destroy the microbes, and this destruction is strongly, we can say, embedded in the memory cell, encoded in the memory cell. So subsequent destruction of the microbes generate the strong memory. The first response is a milder one, that's called the primary immune response. Probably you heard about this term in the textbook, primary immune response. Primary immune response is first time when our immune cells, basically the acquired immune cells, they destroy the microbes, they generate the primary immune response. But subsequent encounter generates the higher intensified secondary immune response. Okay, let me repeat it, it's worth to repeat it. First time when our acquired immune cell destroy the microbes, okay, they generate the primary immune response, which is low intensity, okay, which is low intensity, and the memory built by this low intensity is a low type of memory, we can say. Now in the subsequent encounter of the microbes, our immune cells strongly built a memory about the encounter, okay. So if another time the same pathogens comes, our body is already aware, they know which kind of things they have to secrete so that they can destroy in a first attempt. Understood? They can destroy in the first attempt. So that's why they are called the specific type of immunity. So now, till now we understood the three alternative terms related with this inborn immunity. First one itself called the inborn, second is innate, and third one is non-specific. Whereas acquired, they are also called as adaptive immunity. They are a specific immunity and driven by the memory. Here there is no any memory, okay. The memory is not generated. Clear? Now the another things related with this inborn and acquired immunity is their types, okay, their types. See, all these acquired immunity is basically, all the acquired immune cells is basically formed by the lymphocyte. Now you tell me, what is lymphocyte? Lymphocytes, the cells are derivatives of the immune cells. Acquired immune cells are derivative of the lymphocyte. You know, your body, my body also, we have WBC, one of the type of the we can say the blood. So in the WBC, they are classified, WBCs are classified, okay. Now WBCs are classified on the basis of presence of granules in their cytoplasm. So okay, just recap your previous concept. WBC, okay, WBC, they are classified mainly into two types. One is called the lymphocyte, and another one is called as monocytes. See, these lymphocytes, there are two types. Either we can say T lymphocyte or T cells, and B lymphocyte or B cells. Okay. In the case of this lymphocyte, this T cell and B cell, these two cells, they are functioning as a acquired immune cell. Hope you already studied from the previous classes, you have coming to understand that WBC are involved in the defense mechanism. Now you understood these WBCs, they are classified, and mainly in the case of acquired immunity, these T cell and B cells are working as a immune cells, acquired immune cell, or we can say the adaptive immune cell. Clear? Now, so they are driven by this T cell and B cell. They are driven by the T cell and B cell. So we can collectively call it as a lymphocyte, okay, a type of leukocyte, a type of WBC. Now, for inborn, we have barriers, different types of barriers. These barriers, they are act like both chemical and mechanical barrier, okay. So now what I am going to do, I am taking the innate, or we can say the inborn immune types, and describe their barriers in details, okay, describing their barrier in detail. Hope you all are well, because hope is something which make us alive, channelize, and drive daily our works, okay. So I am hoping you all are well, fine, okay. Now come to the point, we are discussing the types of innate immunity. Types of innate, don't confuse that innate is something new. Innate is inborn, innate, or we can say the inborn, present since birth, inborn. Types of innate or inborn immunity. First types, these are not types, but these are called the barriers, okay. The first barriers in the innate immunity is called physical barrier. It's called the physical barrier, or we can say the physical agent. Physical barrier. Now you tell me, which kind of physical barrier we have? The foremost thing is our skin. The first thing is our skin. Now you are surprising how our skin is protecting us. You know, skin is secreting the sweat, okay, and sweat is salty in nature, clear. They have the less acidity, and due to this acidic and salty power, many of the pathogens won't reside or colonize for a longer period of time in our body, okay. Then we have another barriers like mucous membranes. You know that mucous membrane is present throughout your urinogenital tract, respiratory tract, okay, this oral mucosa, everywhere the mucous membrane is present. What mucous membrane is doing? Mucus is a viscous fluid, okay? It's also involved in the, and you also don't forget your nostrils. Your nostrils also have the mucous membrane, and these specialized epithelial membrane trap the dust particles, the foreign agents, and sometimes microbes too. They are low immune functioning cells, but even though you come across a daily with so many dust, so many foreign particles, so many things, but they are the one who prevent, who strongly prevent by their chemical secretion. For in the case of a skin, you have sweats, and also in the ear we have the ear wax, okay? And in the mucous membrane, the respiratory system, gastrointestinal tracts, all are lined. Nature are so, you know, they are so clever. They made humans, they programmed human in such a beautiful way, where if you maintain your, we can say, the mental and the physical health, you feel awesome, okay? You feel awesome. Now we have the another kind of barrier. We have the another kind of barrier, and that barrier is called the physiological barrier. Physiological barriers. As a name suggests, these physiological barriers are basically the product of our physiology or mechanism of our physiology. Now you are saying what I am saying, work, functions. I am not going to make you confused. It's just basically the secretions. Secretions comes from your GIT. In the GIT, the one specific part called stomach, and the stomach secrete what? What kind of stomach secretion is involved in the immune function? HCl. You know the HCl, hydrochloric acid, they are highly acidic HCl, okay? These highly acidic nature, we can say the enzyme, this highly acidic nature of the enzyme destroy the bacterial cell wall. They dissolve the bacterial cell wall and disrupt the growth of bacteria or microbes which comes along the ingested foods, That's the one point. Second, you know, not always emotional weak person are considered as an immuno weak patient. Why I am saying? Because the emotional persons, they start weeping, okay? इमोशनल पर्सन समटाइम्स वो लोग रोते हैं, इमोशनल हो जाते हैं, आँख से आंसू आ जाते हैं. नाउ द टीयर्स विच इज कमिंग फ्रॉम द आईज, they are important for our immune systems. You know, the tears contain one highly, we can say, the destructive enzymes, and that destructive enzyme is called lysozyme. These lysozyme, they are called the destructive enzyme. Okay, they also won't allow microbial growth in the eyes. Why I am saying the physiological barriers? See, they are restricted along the one physiological systems in our body, like HCl in the stomach, these lysozymes in the eyes. Now you have mouth also. We have mouth also, which we are using like many time. Okay, so mouth. Now what things is present in the mouth? Mouth glands, and they secrete the saliva, and saliva contain antibody. And mainly the IgA class of antibody. You know, 5 class of antibody is present. So IgA type is one type which is present in the saliva. IgA antibody, basically IgA antibody which is present in the mouth. These all, and there are so many others, okay, HCl, lysozymes and these IgA, they are all involved in the immune function because they are restricting the growth of the microbes. That's what we discussed. What is the immunity? We discussed that those immunity is the power of our body which restrict the microbial growth in the body or other foreign particles or any cellular debris. Okay, so they are doing the same job. That's why they are also called as the innate immunity barriers. They are called the innate immunity barriers. Then you understood this? Now we have another barrier called cellular barrier. Our body is like, it's a very awesome, no? If you cross one limit, they make you another limit, then if you cross another limit, then so the another limit. It's not easily get fall, it's not get easily impressed by the microbes. Okay, now another, we have the cellular barrier. We have the cellular barrier. We have the cellular barrier. See, cellular barriers are mediated or done by the three things. You have neutrophils. Neutrophils. You have the other cells called macrophage. And you have monocyte. Now use your brain and tell me what these cells are actually. These are the WBC, types of WBC. You know the neutrophil, basophil, eosinophils, they are the granulocytes, a type of WBC where in the cytoplasm of these cells, we can say in the cytoplasm of these cells the granules is present. That's why they are called the granulocytes. So neutrophils is one of the granulocyte. Neutrophils is one of the granulocyte. Now first we will describe this neutrophil in detail. Why I am describing? Because they have the so many functions which they are doing, which they are functioning, they have the so many diverse functions. Okay, the first, cellular barrier. Don't forget, we are discussing the cellular barrier, and the first one is the neutrophil. Neutrophils. First thing, it's a type of WBC, and more specifically it's a granular WBC or granulocyte. Granulocyte. Okay. Now they are multi-lobed, means suppose this is one neutrophil. Okay, they must have nucleus, otherwise it won't considered as live neutrophil. And see, and other structures like the lysozymes, Golgi body, mitochondria, and other structure is present. Okay, see, initially what happened in the young stage when these neutrophils are produced from, very good, bone marrow. And don't ask me where the bone marrow is present. Okay, so in the bone marrow these neutrophils are producing. Okay, these neutrophils are producing. So when they are produced initially, means their premature state, their nucleus is a single one lobe nucleus. Okay, the nucleus is not divided, but when it's progress, when it start encountering the pathogens or microbes, their nucleus is divided and they make the lobes, and that's are called the multi-lobed. You have to write down, the neutrophils are the multi-lobed cell. It's a multi-lobed cell. That's why they are also called polymorphonuclear cells. Polymorphic neutrophils. They are called the polymorphoneutrophils. Now what happened? These neutrophils are producing from the bone marrow, and they are present in our blood. So what happened? when pathogens comes, they secrete, the neutrophils secrete the enzymes called destructive enzymes, which ruptures the bacterial, we can say the cell wall, bacterial cell wall. The first thing is that. Second thing is they perform one of the immune, we can say the immune function, and that immune function is called phagocytosis. Write down this word, phagocytosis. Now, what is meant by the phagocytosis? Phagocytosis means they engulf, suppose here one microbial antigen is present, by the process of endocytosis, engulfing inside, okay, endocytosis, engulfing inside, by the process of endocytosis, they are capturing, entering inside the cellular environment, and by the secretion of enzyme, these are disturbed, crushed, okay, and released. Understood? That's the one phagocytosis behavior about the neutrophil. Some of the researchers, they shown that when we provide the glucose source, when we provide the sucrose, glucose, fructose to the neutrophils, to the blood, the phagocytosis behavior of neutrophils is down. Okay, in the presence of sugar, the phagocytosis property of the neutrophils is down. And when we are in a fasting stage, okay, when we are in a fasting stage, these neutrophils are so active. They are so active. Okay, maybe it's explained why fasting is considered as a good thing, why they are involved in the rituals. Maybe I'm not clear, but that's a open research, that when you provide the sucrose, glucose to the neutrophils, okay, their phagocytosis engulfing property is diminished or suppressed. But when you are in a fasting stage, the phagocytosis behavior of these neutrophils is enhanced. Clear? Now, they are the short-lived cells. Some of the books write 1 to 2 days, some books write 5 to 6 days, but exactly they are living as 6 to 13 hours only. That's their lifespan. Short lifespan play a crucial role. Clear? Now, another things related with these neutrophils is their conditions. You probably heard about the neutropenia or neutropenia, low neutrophils count. This low neutrophils count is basically seen in the case of chronic inflammation. In the case of the chronic inflammation, they are involved. Why? You must ask why. Always ask why. Now, because they are the one who is going first. I am already told initially, no, that which type of immunity we are discussing? Very good. We are discussing the innate or inborn, and I told it's a non-specific type, means they are blindly going, whatever negative things comes in our body, negative, I mean the pathogens, the microbes, the debris, whatever they are coming, okay, they are blindly going, okay. So they are the one who is going first. These neutrophils are going first as far as the cellular mechanism is concerned, but you know that saliva, their skins, they are the one who is coming first. But as a defense mechanism considered in the form of cells, these neutrophils are the one who is representing the first line management, or we can say the first line presentation towards these microbes is the neutrophils. So in the chronic inflammation, when the inflammation is going on to the prolonged time, these cells are keep on going towards the inflammation site, okay, and eventually they are forming the pus. So in the pus is basically the composition of dead WBC. So in the case of pus, you are going to see lots of neutrophils. In the pus, they are, we can say, the majorly they are present. Majorly in the case of pus, these neutrophils are present. Then another thing, interesting thing about the neutrophils is You know the autopsy or called the postmortem. In the autopsy or the postmortem, when you dissect the heart or brain and see the neutrophils are present, okay, see the neutrophils are present, then what it ruling out? What is the diagnosis that the neutrophils are majorly present in the brain or heart or all things? It shows that patient is either dead as a myocardial infarction or a brain stroke. So, neutrophils is also helping in the autopsy, or we can say in the postmortem. Clear? Now, this is the neutrophils. Now, two things more about the neutrophil. One thing I already told you, it is showing the phenomenon of phagocytosis. Okay, phagocytosis. The another thing it is showing is chemotaxis. Chemotaxis, and one is diapedesis. Phagocytosis you understood, engulfing the bacteria, destroying in their cellular environment, that's leave it. Now come to the chemotaxis. Chemotaxis is they are present, okay, they are present in the cellular environment or inside the cellular space, or we can say the tissue space, they are present, okay, but it have to go in the inflammatory side, injury side. Suppose I have one injury in my hand. It may be the breakage, okay, it may be the fractures, okay, it may be the tearing of the skin. So what happened? Neutrophils have to come on that side, accumulate on that side. So neutrophils are secreting some of the substances, and that substances is called the chemoattractor, which attracts neutrophils towards the injury sites. That's called the chemotaxis. Clear? Now, diapedesis. Diapedesis is basically releasing from the blood and going to the injured area, squeezing out from the blood and going to the injured area. That's called the diapedesis. So about the neutrophils, you keep 2, 4, 5, 3 lines. I don't know how many lines. Keep in your mind is that first is it's a type of WBC. It's a granulocyte. Okay, it's a polymorphonuclear. It performs the three things. First is phagocytosis, second is chemotaxis, and third one is diapedesis. First line presentations towards the foreign particles or the microbes. Clear? Now, come to the another cellular barrier of innate immunity. That's called monocyte. Okay, monocyte is also a type of WBC. Monocyte is also a type of WBC. Now these monocytes, they are not a granule, monocytes are not a granule cells. Okay, monocytes are not a granule cells, means in their cytoplasm the granules is absent. Okay, means they are a granulocyte. One thing. Second thing is that they are the largest. They are the largest WBC. Means long, their diameter is long. And these monocytes, they are differentiated. See, keep in your mind is that they are also producing from the bone marrow. Okay, so I am not giving you again and again the details for their origin. So they are also originated. All these blood cells is mainly originated in our bone marrow. Okay, and by their genetic program, they are differentiated either as a granulocyte, we can say the monocyte, lymphocyte, T lymphocyte, B lymphocyte, eosinophil, basophil, neutrophils. On their genetic programming they are differentiated, but their origin is always the bone marrow. Their origin is always the bone marrow, either a red bone marrow or white bone marrow or yellow, I don't know, yellow bone marrow I think, yeah, red or yellow bone marrow. Okay, now what I am highlighting is that when these monocytes, when they are going into the tissue environment, they are differentiated as macrophages. Monocyte goes into the tissue space and differentiated as macrophages. And you know, macrophages are what? They are the professional phagocytosis cell. They are the professional phagocytosis cell. Macrophages are the professional phagocytosis cell. Now, you know that monocytes is also involved in the phagocytosis activity. They are also involved in the phagocytosis process. Means they also engulf the microbes. They also involved in they engulf the microbes. Okay, you know, in the COVID-19, okay, in the COVID-19, these monocytes are highlighted. They are highlighted because in the case of the COVID-19, the monocytes numbers is also going to be raised. It's also raised in the COVID-19. So they are also one of the diagnostic evaluator in COVID-19. Okay, that's about the monocytes. They are the cellular barriers. Remember, they are the cellular barriers of our innate immunity. Now, the last cellular barrier of our innate immunity is differentiated. What is this? Differentiated. Yeah, don't tell me neutrophils. Differentiated monocytes. That's the macrophages. Macrophages. There are lots of ongoing studies on the macrophages because they are called, I already told you, no, they are called professional phagocytosis cell. Professional phagocytosis means they are the one who is primarily or purely, we can say, involved in the engulfing of cellular debris, antigens. Okay, another thing we can say, the viral antigens, viral proteins, everything they ingest. They are very powerful phagocytosis cells. They are the very powerful phagocytosis cells. Okay, now these macrophages, they engulf and digest the pathogens, okay, such as the cancerous cells. You know that they are the differentiated monocytes. Since their names are differ according to the presence in particular or specific area. You know, in the case of liver, we have one cell called Kupffer cells. Kupffer cell is what? Kupffer cell is basically the macrophage. Write down Kupffer cell. Kupffer cells providing the phagocytosis activity or providing the innate immunity in the liver. Okay. Then we have microglia cells present in central nervous system. Okay. Then we have osteoclast. osteoclast. Where osteoclast is present? Yeah, it's present in the bone. Okay, it's present in the bone. And we have Langerhans cell. Langerhans cells present in our pancreas. So all these Kupffer cell, microglial cells, osteoclast and Langerhans cells, what they are? They are the macrophages, which involve primarily in the engulfing of the bacteria. If I am talking it in detail, describing in details, it secretes a phagosomes. It secretes a phagosomes, okay, and by the secretion of these phagosomes, the microbes are trapped. The microbes are trapped inside, and they are destroying in one sequential manner, by the destructive enzymes, in the lysosome of the macrophages, They are also called anti-inflammatory cells, because they are against the inflammation. Clear? They are also called antigen presenting cells, because they are the one who give message to the acquired type of immunity, T cells, to come. We are no longer last. We are not like winning the match. You come and support us, that's why we win. So they are also we can say the presenting the T cells. So these all are the cellular barriers. Now the last one is called cytokinin barrier. This cytokinin is basically the specialized proteins. They are the specialized proteins which act as a immune functional cells. Last one is called cytokinin. These cytokinins is basically the protein. Okay, these are the proteins which secrete in the form of interferons, which destroy viral affected cells. You know that once viruses come in our body, okay, and viruses once comes, they interfere, they compare and contrast, they make the fight and colonize themselves in our cellular machinery. They capture the cellular machinery, okay, they rupture the cellular machinery and they make cell die. So what happened? Those cells who are affected by the virus, these cytokinins, they secrete one strong proteins called interferons, which destroy those viral affected sites and prevent further viral infections. Okay, these all are the innate immunity, or we can say the inborn immunity. So for concluding today class, we discussed about the immunology. That immunology is the study of the immune system, their normal function, abnormal function, interaction with various substances called the drugs. Okay, then we seen different branches of the immunology, like the diagnostic immunology, classical immunology, immuno-oncology, psycho-neuroimmunology, okay, and so forth. Then we discussed about the immunity, that immunity means restricting the pathogens to come in the body and manifestate their behavior. Okay, then we defined the immunity system, okay, and classified immunity system. Always remember one word: immunity systems is the one who knows what is our self and what is our non-self, means what is my own and what is my not own. Okay, they are the masters in this, making the differentiation between the self and non-self. Clear? Then we studied about the classification. The first classification we seen is the
innate or inborn immunity, or also called the non-specific immunity, and then their types, the barriers, all those 4 barriers. Then in our further lectures we will cover about the acquired immunity and discuss